All right. Good evening, everyone. I hope you're all doing well and getting through finals week. You're almost there, right? Just a couple more days. So, there are a lot of questions in this review, so we will go ahead and get started because I think I have like 70 or so questions in here. So, let's go ahead and try out the heart, shall we? So, first things first is the first section we covered in AP2 was the heart. So, the ventricles pump blood into these two major outflow vessels. So we have the right ventricle and the left ventricle. Where do they pump blood to? So the right ventricle is going to eject blood and send it into the pulmonary trunk. And the left ventricle is going to eject blood and send that into the aorta, right? So the pulmonary trunk, we can see there is coming off of the right ventricle here. And then we can see that the left ventricle is sending its blood into the aorta. All right. So the right atrium receives blood from these three vessels. So two really big vessels and one critical but smaller vessels. What vessels supply the right atrium with deoxygenated blood? Yep. So the superior vena cava and the inferior vena cava are the two major vessels that we think of, but then we have to remember that there's coronary circulation as well, and so that coronary sinus is also going to be supplying blood. So superior vena cava draining blood from the head and the arms, the inferior vena cava draining blood from, you can just think of like everything below the diaphragm. And then you really can't see it in this picture, but that coronary sinus would be draining blood in about here-ish. So we'll just kind of draw a dot for it. So those three vessels are all supplying deoxygenated blood to that right atrium. All right, let's trace a drop of blood from the superior vena cava to the aorta. So we're going to go from the superior and inferior vena cava. We're going to go into what we just saw, which was going to be the right atrium. And then from the right atrium, where are we headed off to? So from there, we're going to head into the right ventricle. And in order to get into the right ventricle, we're going to have to pass by the tricuspid or the right atrioventricular valve. Then we're going to be in the right ventricle. We saw in the first question that from the right ventricle to pass into the next place that we're going to, the right ventricle supplies the lungs with blood. We're going to work our way into the pulmonary trunk. And then to get into the pulmonary trunk, we have to pass through the pulmonary semilunar valves. Then once we're in the pulmonary trunk, we are going to then pass through the pulmonary arteries, and the pulmonary arteries will get us to the lungs. Then to get back to the heart from the lungs, with oxygenated blood, we are going to use the pulmonary veins. And the pulmonary veins are going to take us back to the left side of the heart. And on the left side of the heart, we are going to find the left atrium, which is where we're going to start adding blood into. And the blood will then work its way into the left ventricle. And in order to get into the left ventricle, we're going to have to pass through that bicuspid valve or that left atrioventricular valve. And then from the left ventricle, we're going to get ejected into the aorta. And in order to get blood to go from the left ventricle into the aorta, we'll bypass that aortic semilunar valve. And then we're going to work our way through the body. We'll see some arteries, and then we'll reach capillaries where exchange will happen. And then veins will take us back to the superior and inferior vena cava. So big pathway, but hopefully it all sounds familiar. All right, so now let's trace the route through the heart, but let's think about the conduction system. So what makes your heart beat? What is the thing responsible for telling your heart to beat? Yeah, the SA node is going to be responsible for telling the heart to beat. Then after that, we're going to have the atria contract. And then what's next in that system? After the SA node and the atria contract, who is next that's going to tell, yep, the AV node, the atrioventricular node. Then that AV node is going to lead into the AV bundle, which makes sense. It's also called the bundle of His. And then we're going to split into a left and right what? Yep, bundle branches, perfect. And then the bundle branches are going to stretch far out into the ventricular myocytes as Purkinje fibers, right? And that's how we're going to tell the ventricles that it's their time to contract. All right, what does the P wave represent in an ECG, and what a mechanical event does it lead to? So what does the P wave tell us? Yep, it tells us that the atria are going to depolarize, and by depolarizing the atria, what do we tell them to do after that? Perfect. Then after that, we're going to have atrial contraction. So keep in mind that the P wave is this first thing here. And then after that, we don't really see anything, right? This PR segment that we see after that is going to be the atrial contraction. And it doesn't look like much, but that's because the ECG is a measurement of electrical activity. And so we don't get to see the physical stuff. We just know that it's happening during that segment. Same thing is true of the ventricles, where QRS would be where we have our ventricular depolarization. And then we wouldn't actually get to see the ventricular contraction in the ECG. We just know it was happening during that ST segment. All right, what causes the opening and closing of heart valves? So if you needed to open your semilunar valves, what would make that happen? Yeah, so pressure would be the big thing that would cause that, right? So pressure pushes open the semilunar valves and allows for blood to pass through. Pressure is what pushes the AV valves closed. All right, so up next is blood vessels. So true or false, arteries carry oxygenated blood and veins always carry deoxygenated blood. Yeah, that's going to be false. So what would be an artery that carries deoxygenated blood? Yeah, so the pulmonary arteries would definitely be arteries that carry deoxygenated blood, and then the pulmonary veins would be veins that carry oxygenated blood. So what we can say always about is that veins always carry blood back toward the heart, and that arteries always carry blood away from the heart. So the oxygenated deoxygenated works out most of the time, except for these pulmonary exceptions. But arteries always carry blood away, veins always carry blood back towards the heart. That we can say with certainty. All right, describe the structure of a capillary. So what is a capillary made of? So capillaries are going to be really small, right? They're going to be a very thin membrane, so they're only one cell layer thick, and that one cell layer is going to be simple squamous epithelium. And so we're just going to have that one very thin layer, and then they are also going to have a basement membrane around the outside of them. So given that they're only made up of this endothelial layer, the tunica intima, and the basement membrane, what is the relevance of this? Why do they have such a thin wall as opposed to something like arteries or veins? Yeah, it's because capillaries are the only place where we can do exchange at. Arteries are just there to get the blood to the capillaries. That way we can have exchange. But the only place that we can effectively have any kind of exchange is the capillary. Veins just help to get the blood back, right? So we need these very thin walled capillaries because that's where exchange happens at. All right, name the three vessels that branch off of the aorta. Oh, so gas exchange happens, but we're also going to have other substances being exchanged as well. So this is going to be where we supply nutrients to the tissues. This is also going to be where we take up waste from the tissues as well. So you can imagine that we have all these things in the blood, sugar, all this other stuff. It's going to be able to, small things will be able to come out and be supplied to the tissue, and then we'll take up things like waste, so like metabolic waste products. So we tend to just talk about gas because we focused on the respiratory system for a while, but this would also be for nutrients and waste too. Only small things though, right? We're not going to have like, you shouldn't be having like red blood cells coming out or like really big proteins or anything. So small stuff is able to leave. Yeah, so we have the brachiocephalic, we have the left common carotid, and the left subclavian. So keep in mind that when we think about blood vessels, there is only one brachiocephalic artery, right? So we only have one of these, whereas we have two brachiocephalic veins. Then after that, we have the left common carotid and the left subclavian. And the way we can remember that is to think of the A, B, C's, right? Brachiocephalic is the B, common carotid is the C, subclavian is the S, and ABCs. All right, what is the purpose of the hepatic portal system? So why do we have this portal system that has to do with the digestive system and the liver? What is the overall purpose of that? Yeah, because we absorb a lot of things from our digestive system, and there is the potential that the things that we absorb are not in the state that we'd want to be in, or that we don't want them to be sent all throughout our cardiovascular system. So the portal system there is responsible for taking up what we've absorbed from our digestive system and then allowing us a chance to inspect that or kind of look over that material. So keep in mind that a portal system is where we're going from a capillary bed to another capillary bed. So we're going from capillary beds in the digestive system and then heading to capillary beds. They're going to be in our liver. And that way we absorb things in the first capillary bed, the one that's in the digestive system, and then we inspect and review those things in the second capillary bed in the liver. So the whole purpose of this hepatic portal system, this capillary leading to another capillary, is allowing us to look over the things that we've absorbed from our digestive tract to make sure that they're what we want to send throughout the rest of our cardiovascular system. You can think of this as like the digestive system customs, right? A lot of stuff coming into the body, getting absorbed, and you want to make sure that there's not going to be anything, like invasive species, like pathogens, we could think of it that way, right? Or other things that aren't allowed, right? No unpasteurized cheeses or something like that. That's what I think of when I think of what customs looks for. I'm sure it's more complex than that. That makes sense to everybody? All right, if that sounds good, then let's talk about hemodynamics. All right, in which blood vessel is pressure the lowest, and in which blood vessel is pressure the highest? Yeah, so it's going to be highest in the arteries and it's going to be lowest in the veins. And that makes sense because where is the pressure generated? It's generated at the heart, right? And everything has to go from high pressure, which is going to be the beginning of this graph, to low pressure, which is the end of this graph, because we're going back to the heart. This is the circulatory system, right? So everything's going from high to low. It makes sense that the pump of the system, the heart, the left ventricle, has the highest pressure. So the arteries then get that highest pressure. It also makes sense is that we work our way through the cardiovascular system. The pressure just has to keep decreasing because we're moving from high to low. And that means the lowest pressure has to be right before we load that blood back into the heart. And that's going to be in the veins, specifically the vena cava. So the highest pressure we'd see in a vessel is in the aorta, and the lowest pressure we'd see in a vessel is in the vena cava. And that's just because blood flows from high to low, and high pressure comes from the pump, the left ventricle. All right, what helps veins get blood back to the heart? What improves venous return? Three things. Valves, absolutely. One-way flow, right? Unidirectional flow. Skeletal muscle pump, perfect. Veins are not going to be able to stay open when a skeletal muscle around them is contracting, so it just squeezes them shut and pushes blood forward. That works real well with the valves because the valves allow for one-way flow. And then finally, the respiratory pump, right? Every time you change your thoracic cavity volume. So if you take a big deep breath in and you increase the volume of your thoracic cavity, you decrease the pressure within it, and that allows for you to push blood. It makes it easier for that blood to move into your thorax because low pressure is easy. You go from high to low, and if there's lower pressure in the thorax, that's a great place for the blood to go because now it has a lower pressure. So the three things are valves, skeletal muscle pump, and the respiratory pump. All right. In which blood vessel does blood move the slowest? So who has the lowest blood velocity? Yeah, the capillaries have the lowest blood velocity. Why is that physiologically favorable? Why do we want to make sure that the capillaries have low blood velocity? Awesome, spot on. So we want this because the capillaries have to do exchange. When we think about arteries, arteries can conduct blood really quickly because that's their purpose, right? The big arteries are called conducting arteries. So it makes sense that sending blood through them rapidly would be a good thing for them. But by the time we get to the capillaries, that's going to be where we need to have exchange at. And so we want to have blood velocity there the slowest. If you want the physics behind it, it's because of what's happening with cross-sectional area. So we think of capillaries as being really small, and they absolutely are, but we have a ton of them. So a billion of these tiny things is going to give us a lot of surface area. And since we're having to take all this blood and put it through all this big surface area, it makes the velocity really slow. The big picture here is the lowest blood velocity, the place where your blood is slowest, is in the capillaries, and that's good because it gives you time for exchange. Everyone feels good about that? All right. So what does systolic pressure measure and what is diastolic pressure? And then knowing what these things measure, what is a normal blood pressure? Yeah, so systole in the heart is when we have contraction. So systolic pressure is the higher pressure because of contraction. Diastolic is when we're in diastole or relaxation. So that's going to be a lower pressure. So then what's a normal blood pressure? If you go to your physician or I don't know, even go to the grocery store, right, and put on one of those cuffs. Yeah, 120 over 80 millimeters of mercury. So I have a lot of detail on the right, but the big picture here is 120 over 80 millimeters of mercury. The 120 is the systolic and the 80 is the diastolic. Yeah, when I'm saying contraction, for specifically for the systolic pressure, what's generating systolic pressure is contraction of the left ventricle. The blood pressure in your body, in terms of systolic pressure, is dictated by left ventricular contraction. The force at which it contracts is going to give you all the pressure you have for systolic pressure. And so we can see that in the picture here, right? 120 is the high pressure. Down here, the low pressure is 80. So up here is systolic. Down here is diastolic, right? And this is just showing us how we take a manual blood pressure. So systolic pressure is the pressure that is going to be the high pressure because of left ventricular contraction. Diastolic is going to be how low of a pressure we reach in between ventricular contractions. 120 over 80 millimeters of mercury is a great normal value to have for that. All right, what is mean arterial pressure? So just from the name, we can make a pretty reasonable assumption, right? Mean is average. So it's the average pressure in your arteries, right? So it's giving us the average pressure in the overall cardiac cycle. So we're saying, what is the average pressure we have in the arteries throughout a cardiac cycle? And this is important because we already said we have a high pressure, right? That's systolic pressure up here. And we have a low pressure down here, that diastolic pressure. And so there's this temptation to just be like, oh, systolic pressure is 120, diastolic pressure is 80. So we'll just do, add them together and average them. But that doesn't work out because we don't spend the same amount of time in systole as we do in diastole. When you're sitting there at rest, you're probably spending 1/3 of your time in systole and 2/3 of your time in diastole. So we need a little bit more complexity in how we calculate this. But the big picture is that mean arterial pressure is the average pressure during the cardiac cycle. So it is just what is the average pressure in your vessel. And in this particular picture, it's this gray line here is where they're getting at that average pressure. So if you're sitting there at rest at 120 over 80, your mean arterial pressure is going to be about 93 millimeters of mercury. And if you want another calculation, right, it is diastolic plus 1/3 pulse pressure. Ooh, I am terrible at writing with a mouse. But that says diastolic plus 1/3 of pulse pressure. And the reason why it's closer to the diastolic value or the diastole value is just because you spend more time and at rest than you do during systole. And that's just because you're in the rested state for the most part. All right, so polycythemia, which is going to be where you have an increase in red blood cells, would have what effect on blood viscosity? So if your body started producing a bunch of extra red blood cells, what would that do to the viscosity of your blood? Yeah, your blood would become more viscous, right? So we can kind of like, I'm gonna exaggerate this, but we can make sense of this by thinking about, if you had to drink water through a straw, you'd be like, oh, that's no big deal. I do that all the time. It's because water isn't very viscous. But if I handed you a cup of honey and I was like, hey, drink this honey through a straw, that would be substantially more difficult and it would require a lot more suction pressure in order to move that honey. And that's because the honey is more viscous. So that's why we'd want to know about this, because polycythemia is making your blood more viscous, which means it's going to require more pressure to push through the system. All right, what three factors would decrease blood pressure? Or you can just think of like what three factors regulate blood pressure, And then how could we change them to decrease blood pressure? Yep, cardiac output is the first one. So if we wanted to decrease blood pressure, would we increase or decrease cardiac output? Yeah, we'd want to decrease cardiac output. And so if we remember that cardiac output is equal to heart rate times stroke volume, decreasing either one of these, either heart rate or stroke volume, or both of them, would be a way to decrease our blood pressure. Perfect. We could also decrease our resistance, right? The resistance is going to be the diameter of the blood vessel. So if we increase the diameter of the vessel, that is going to do what? Decrease resistance. We could also technically decrease vessel length. That's not really something that we're going to be it's not a major thing we're going to do, right? Technically, we'd have to like lose an appendage. So that doesn't sound like a great way to do that. We could also decrease blood viscosity, which we just talked about in the previous example, because we saw a way to increase blood viscosity. Then the last thing is blood volume, right? That was the big thing that we focused on with kidneys because they regulate your urine output. If your kidneys are allowing for a lot more fluid to leave, then you're going to lose that fluid. You're going to decrease your blood volume, and that's going to decrease your blood pressure. So the three factors that regulate blood pressure are cardiac output, resistance, and blood volume. And decreasing any of these is going to decrease your blood pressure. So a decrease in cardiac output, a decrease in resistance, or a decrease in blood volume will decrease your blood pressure. How we do that, it depends on which factor, right? Decreasing heart rate and stroke volume would be a way to decrease cardiac output. Increasing vessel diameter would be the major way if we think of decreasing resistance, and decreasing blood volume would just be losing fluid. All right, what stimulus does the renin-angiotensin system respond to, and how does it reestablish homeostasis? So more importantly, the effector thing here is angiotensin 2, right? What does angiotensin 2 tell you to do? Yeah, angiotensin 2 tells you to constrict. It tenses your angios. It tells you to vasoconstrict. And that means it tells you to increase your blood pressure. Since it's a part of the renin angiotensin system, essentially what it's getting at is it'll help you to deal with low blood pressure. If the effector thing here is going to be angiotensin 2, then it's going to raise your blood pressure when you have low blood pressure. So this is good for you if your blood pressure is low because Ang2 increases your blood pressure. So if your blood pressure gets low, since it's a negative feedback system, you will increase your blood pressure in response to it. So Ang2 raises your blood pressure. All right, so then what about atrial natriuretic peptide? What does atrial natriuretic peptide do? If angiotensin 2 increased blood pressure, what does atrial natriuretic peptide do? Yeah, it decreases blood pressure, and that's because it allows for us to get rid of excess fluid. So it's going to promote the loss of salt. That's what the natriuretic thing means. And it's going to also result in water loss. So ANP is going to allow for us to decrease blood pressure. So let's just kind of put this in the context of all three of these hormones and put them together real quick. So we have angiotensin 2, we have AVP, ADH, and we have atrial natriuretic peptide. So let's just make sure we know what all three of these do. So what do we say angiotensin 2 does to blood pressure? It increases it, right? What does vasopressin antidiuretic hormone do to blood pressure? Yeah, it increases blood pressure as well. And then what does atrial natriuretic peptide do to blood pressure? It decreases it, right? So two of these increase blood pressure, one of these decreases blood pressure. Angiotensin 2 and vasopressin increase BP, atrial natriuretic peptide decreases BP. All right, so then that gets us on to blood, unless anyone has any questions so far. I know I've been going through stuff quick, but I made a lot of questions, so. All right, so what are the formed elements and what are their functions? So first of all, just what are the formed elements, right? Three things. Yeah, red blood cells, erythrocytes, white blood cells, leukocytes, platelets, thrombocytes. Perfect. What do they do? What do red blood cells do for you? Yeah, red blood cells do gas transport, right? What do white blood cells do? Yeah, immune stuff is good enough, right? Immune stuff. And then what do platelets do? Blood clotting, right? Or, the fancy name of that would be hemostasis. So red blood cells do gas transport. White blood cells do immune stuff. It's complicated, right? And then thrombocytes or platelets are just going to be responsible for blood clotting or hemostasis. All right. So what is hemoglobin and what does it do? Yeah, so hemoglobin is the structure inside of red blood cells that's going to be responsible for gas transport, right? So there's two parts to that word. There's heme and there's globin. And there are four heme molecules, which means that we can carry four oxygens per heme. I'm sorry, we can carry four oxygens because there's four hemes. There's also the globin, and carbon dioxide can bind to the globin. So the heme is going to have the iron, which binds the oxygen, and there's four of them. 1, 2, 3, 4, and that's where we're going to bind oxygen. Then the globin can bind carbon dioxide. It's not the best at it, but it does a reasonable job. All right, where do platelets come from and what do they do? Yeah, so they come from something called megakaryocytes, which are going to be found in the red bone marrow. And the platelets' job is to help with blood clotting. Specifically, in the process of hemostasis, they help with one of the steps in that. So they are not cells. It's a critical thing to know. So the megakaryocyte is a cell, but then the megakaryocyte tears off pieces of itself, and those cell fragments are the platelets. So megakaryocytes just remain in the bone marrow. And then the pieces of the megakaryocyte come out, and those are going to help us with clotting. So these pieces of the megakaryocyte are the platelets, and the platelets help us with blood clotting by working their way through the blood and then sticking together when we need them to plug a wound. All right, so let's think about hypoxia and the negative feedback loop associated with it. So if you were somewhere that had a low amount of oxygen, so maybe you went to like the top of a mountain or something like that, right? So if you went skiing for the weekend and you were on top of this mountain and there was a low amount of oxygen, what would your body want to do to compensate for the lack of oxygen? Yeah, you'd want to increase the amount of red blood cells that you have. And that makes sense, right? And the way that we'd increase the amount of red blood cells that we have is by producing more EPO, which comes from the kidneys, and that would tell us to make more of those red blood cells. So what would that do? When we have all these red blood cells, what would we be better at doing? Yeah, so hypoxia is a decrease in oxygen. So you have less oxygen being transported. Since you have less oxygen being transported, you're going to sense that and then release EPO, which tells you to make more red blood cells. And red blood cells are responsible for transporting oxygen. That would give you more ability to transport oxygen and help you to better deal with this environment that is hypoxic, that doesn't have enough oxygen for the amount of red blood cells that you had. Perfect. So this is negative feedback, right? Low oxygen transport. We increased the amount of the cells that could transport oxygen, so then we restored our oxygen carrying capacity. All right, so we want to work our way through all of the 5 white blood cells, and then are they granular or agranular? Yep, so the granular ones are going to be neutrophils, eosinophils, and basophils. Perfect. And they all have fill in their name, so they are filled with granules. Then the other two are monocytes and lymphocytes, and they do not have fill in their name. They are not filled with granules, so they are agranular. So if they have cyte in their name, they are agranular. If they have fill in their name, they are granular. So now let's make sure we know what is like the hit song of each of these white blood cells. They all do a lot of stuff. Let's just know the major thing that they do. So neutrophils, what are they responsible for? Yeah. Bacterial infections. Eosinophils. If you saw an increase in them, what would you think of? Yeah, parasitic infections. Basophils, what do we think of? Allergies and inflammation. All right, monocytes. What do monocytes grow up to become? Yep, they end up being macrophages. They help us phagocytize things. Then what do we think of with lymphocytes? They do a lot of stuff, but what is like the hit song thing that we think of? Yeah, so immune cells, antibodies, sounds good, right? They do a lot more than that. They do a lot of stuff with specific immunity. They just do a lot of stuff, but we just want to know the one major thing. So we'll think of like antibodies and more specific immunity. Perfect. All right, so let's make sure we know the 3 steps of hemostasis. What are the 3 steps of hemostasis in order? Yeah, so the first thing we're going to do is vascular spasm. All this is just constricting the blood vessels. So if you just constrict the blood vessels a whole bunch, that is going to reduce the size of them and therefore reduce the amount of blood that they lose. Then after that, we're going to form the platelet plug. This is where the platelets or the thrombocytes get involved. They are going to help us to form this temporary platelet plug. And then finally, we're going to work through the more complex path of coagulation. And the whole purpose of this is so that we can get to the point to where we have fibrin. Fibrin is this molecular rope that we're going to use to just tie all the stuff together. Anything that we have, red blood cells, platelets, white blood cells, whatever we have, just tie all the junk together and use that to fill in the hole, the damaged area. So after an injury, we do 3 things: vascular spasm to reduce the amount of blood loss, platelet plug, a temporary way to stop the bleeding, and then coagulation, a more permanent way to stop the bleeding. And that's with fibrin. All right, a person with anti-B antibodies would have what type of blood? Yeah, a person with anti-B antibodies could have A blood. What else could they have? Yeah, they could also have O blood, right? So if they just had anti-B antibodies alone, then they would definitely be A blood type. But if they had anti-A and anti-B antibodies, then they could be O blood type, right? All right, a person with AB blood can receive a transfusion from what other people? Who could give blood to someone with AB blood? Yeah, someone who has AB blood could receive blood from everyone in terms of ABO blood typing, right? Why is that? Why could someone with this blood type receive blood from anyone? Why are they the universal recipient? Yeah, they don't have antibodies against antigen A because they have it. They don't have antibodies against antigen B because they have that too. And then they're not going to have antibodies against anything on O because O doesn't have either of these antigens anyway. So this weird picture here is basically just a donation scheme. AB can donate to itself. It can receive blood from A. It can receive blood from O. It can receive blood from B. So a person with AB blood could receive any of these, and that's because they don't have antibodies against any of this stuff, right? They are happy to receive blood from any of these people. All right, what makes someone's blood type A positive or A negative? What is this positive and negative stuff? Yeah, so that has to do with the D antigen or RH. And so it's whether or not they have this. Just like if someone is A, whether or not they have antigen A, with positive or negative, it's whether or not they have antigen D or they have this RH factor. So if someone is positive, they have antigen D, they have RH. If someone is negative, they do not have them. And the whole reason why we've separated this out from the other types of blood is just because you don't start life with antibodies against RH. If you are negative, you would have to have exposure to the D antigen to create antibodies against it. So that's why it gets separated out. But the basics of this is just the same. If you are negative, you do not have the D antigen. And if you are positive, you do have the D antigen. So your red blood cells will be expressed with it. It's just that there are no naturally occurring antibodies. So you're not born with antibodies against this if you are negative. And if you're positive, it doesn't matter. All right, so let's work through all of these blood types. So sample 1, what blood type are they? O positive, perfect. What is sample 2? O negative. Sample 3, clotting in the first well and in this third well, the anti-A. All right, A positive. Sample 4, just clotting in the first well, A negative. Sample 5, clotting in the anti-B and the RH wells, B positive. Sample 6 is B negative. Sample 7, clotting everywhere, AB positive. And sample 8 is AB negative. Awesome. Looks like everyone did well on their lab exam, right? All right, lymphatic system. So, sorry, got ahead of myself. What is lymph? Or what is lymph suspiciously similar to? Yeah, so lymph is definitely going to be interstitial fluid, right? And a good portion of the interstitial fluid is just going to be what gets pushed out of the plasma. So lymph is definitely just interstitial fluid that is pushed into lymphatic capillaries. But that interstitial fluid, or a decent amount of it, is going to be coming from the plasma. And so what's happening here is the same thing that we think of as, like blood moving or anything else, right? Things are going from high to low, and the place with low pressure is in this lymphatic capillary. So the interstitial fluid is going to get pushed into the lymphatic capillary, and then some of the plasma will be pushed into the interstitial fluid. So the lymph is going to be really similar to plasma, right? The biggest difference is going to be location. I mean, there'll be a little bit more waste products, things like that in the lymph. But the big thing we want to think of is just, that this is going to be primarily a location change. The one exception to this would be in the digestive system, because we're going to have lacteals. And so we're also going to have fat included in those lacteals. So lacteals are just lymphatic capillaries that are in the digestive system. And since the digestive system is absorbing fats, some of that fat ends up going into the lymphatic system. And so it gets absorbed into those lacteals. All right. How do lymphatic capillaries compare with blood capillaries? What is going to be the major difference? So when we think of a blood capillary, we tend to think of it being, we have an artery connected to a capillary, connected to a vein. Yeah, the lymphatic capillaries, spot on, are going to be closed ended. So they're not going to be attached to something at that end, right? The closed end is just hanging out near the blood capillary. And that's going to be important because that's why we need to be taking up things from the interstitial fluid. That's why we're having this plasma, this high to low pressure gradient. And then once we put things into these lymphatic capillaries, unlike the circulatory system where things have to move very quickly so that we can get the blood back to the heart to pump it out again, things will move a lot slower, right? I think of the lymphatic system kind of like the lazy river. So things will move slow, and then it's going to allow for processing in those lymphatic nodes. So a chance to look things over and make sure there's nothing that we need to mount an immune response against. All right, so how do lymphatic capillaries compare to veins? What's similar between lymphatic capillaries and veins? Yeah, they both have valves. They're both going to be low pressure as well. They can both benefit from the skeletal and respiratory pumps because they are easy to compress, right? And then they benefit from that low pressure in the thoracic cavity. They actually end up being both composed of the same three tunics. So differences would be veins get to benefit from the high pressure of the heart, right? So the veins are going to have that heart to help move blood, whereas the lymphatic system doesn't have that benefit. And then the lymph nodes are going to be found in the lymphatic pathway, but we're not going to have lymph nodes in our veins, right? All right. So what are the two lymphatic ducts and what drains to them? Or rather, what are the two lymphatic ducts and which one does more work? Yeah, so we have the right lymphatic duct and then we have a left lymphatic duct which is called the thoracic duct. And between them, the thoracic duct is doing the vast majority of the work. If we look at this picture here, everything in purple is what is drained by the right lymphatic duct, and then everything in yellow is drained by the thoracic duct. So the right lymphatic duct really just takes care of the right side of the head, the right arm, and the right part of the thorax. Everything else, both your legs, your pelvis, the majority of your abdomen, and everything else on the left side is all drained by that thoracic duct. So there is definitely an uneven distribution of work here. So the thoracic duct does the majority of the work. All right, so we have innate and adaptive immunity. What is innate immunity, or what is an example of it? Yeah, the skin is a great example of it. And what does the skin protect you against? Yeah, the skin protects you against everything, right? So there's a bunch of examples here. I wouldn't expect you to know all these examples, right? But the important thing is here is that these protect you from everything. The skin doesn't need to know anything about what it's protecting you against. It protects you against everything. It is a physical barrier. Having a low pH in your stomach protects you against everything. The low pH doesn't care what type of bacteria you are or what type of pathogen you are. It's going to just protect you against everything. So innate immunity protects you against everything without knowing anything about it. This is like walls or like doors. They protect you from everything. They don't need to know anything specific about it, right? Doors are good at protecting you from, I don't know, like mosquitoes, but they're also good at protecting you from coyotes, I guess, right? They protect you from everything. So innate immunity protects you from everything. It doesn't need to know anything about it. All right, so respiratory system. Which respiratory structure is a passageway for both food and air? Yep, the pharynx, right? Specifically in the pharynx, it would be the oropharynx and laryngopharynx, but the pharynx in general is a good answer because it does have food and air passing through it. All right, where is the epiglottis found and what is the function of the epiglottis? So, what organ is the epiglottis a part of, and then what does it do? Yeah, it's part of the larynx, and its job is to make sure that food is only able to pass into the esophagus and that air passes into the larynx and down the rest of the respiratory tract. So it makes sure that you don't end up with food or liquids in your respiratory tract, and make sure that the food and liquids do pass through your digestive tract. So food and liquids, esophagus, air in the larynx, and then down to the um, trachea. All right. If the pressure in the atmosphere is 760 millimeters of mercury, what does the pressure inside the lungs need to be in order for inhalation to occur? So if I want air to go into my lungs, what do I want the pressure to be inside my lungs? Yeah, anything below 760 will work. So 759 will work, 758 will work, anything lower than 760 because the air goes from high to low, right? Just like everything else. So anything below 760 is good. All right. So how then do we decrease pressure in the lungs? If I want to bring air into my lungs and I need to get to this pressure that's lower than atmospheric pressure, I need to do something to my lungs, and that something is increase the volume. So if I want to increase the volume, that will decrease the pressure. And so what we can see here is in inspiration, I am contracting my diaphragm and contracting my external intercostals to increase the volume. Increasing the volume decreases the pressure. Low pressure makes air go into my lungs. All right. What is partial pressure? Yeah, so partial pressure is the pressure of an individual gas in a mixture, right? And that's important because when we think about in the lungs, are carbon dioxide and oxygen going the same direction? No, right? They are definitely not. So when we think about in the lungs, oxygen is going from the alveoli to the blood, but carbon dioxide is going from the blood to the alveoli. And the only way we can know that is because of partial pressure, because we can measure the pressure of these individual gases. So we need partial pressure so we can see what individual gases do. That way we can understand gas exchange, and we don't just say air is going somewhere, right? All right. What is the difference between lipid soluble and water soluble hormones in terms of where would they bind? Where would a lipid soluble hormone be able to bind? Where would a water soluble hormone be able to bind? Yeah, lipid soluble hormones can pass through the lipid membrane, and so they'll bind within the cell. Water soluble hormones can't pass through the membrane, and so they have to bind on the membrane itself. So the pictures have a lot going on here. Don't worry about that. The important thing is this lipid soluble hormone goes into the cell and binds to a receptor inside the cell. The water soluble hormone can't pass through the membrane, so it has to bind on a receptor on the surface of the membrane. Lipid soluble can go inside, water soluble has to stay outside. All right. So there were three ways that we could regulate hormonal release. What were the three ways that we could regulate hormonal release? Yeah, we could have a hormone leading to the release of another hormone. This was like how the anterior pituitary worked, right? So a hormone could lead to the release of another hormone. We could have a change in the blood. It had a very fancy name called humoral stimulation, but this would just essentially be a change in the blood. So an increase in blood glucose leads to the release of insulin. And then we could also have the nervous system telling us to release a hormone. And this is how like your adrenal gland works. So what could tell you to release hormones? Other hormones, the nervous system, or a change in the blood. What part of the brain regulates the secretion of hormones from the pituitary gland? Or who's in charge of the pituitary gland? Yeah, the hypothalamus is going to be in charge of it. So you could even see that the pituitary is like hanging off of the hypothalamus. So the hypothalamus sends messages and tells us to release things from the pituitary. So, and then in terms of the pituitary, the anterior pituitary releases something called the adrenocorticotropic hormone, ACTH. What organ in the body is ACTH sending that message to? Yeah, exactly like the name suggests, right? So adrenal, it is sending it to the adrenal gland on top of the kidney. And then it tells you to release cortisol, a hormone leading to a hormone. All right, what comes from the posterior pituitary? What two hormones? Yep, vasopressin, so retaining water, and oxytocin, which is responsible for milk letdown and responsible for labor and delivery. So vasopressin and oxytocin come from the posterior pituitary. Anterior pituitary are those flag top hormones. All right, digestive system. What is the second phase of digestion in the stomach? First, we have the cephalic phase, and then what is the next phase? Yeah, the gastric phase. And what do we do in the gastric phase? What is the stomach doing during that phase? Yeah, it's doing stomach stuff, right? It's secreting things like gastrin to tell it to do stomach stuff. It's increasing motility. It's going to be releasing all of the typical things we think of, secreting things that allow for digestion. So we're doing stomach stuff. We're actively digesting because there's food in the stomach. All right. So, what is active in the stomach? Since we just said we're doing stomach stuff, what kind of enzymes are going to be active? What is going to be released into the stomach? Yeah. So, pepsin is going to be active. What does pepsin help us to digest? Yeah. Pepsin helps us to digest proteins. Perfect. What else is going to be active in the stomach? Any other enzymes? Yeah. We have a lipase that's going to be active there. Perfect. Yeah. So, we have a gastric lipase that's made by the stomach that's going to be secreted and active there. But we also have lingual lipase, which gets activated there. All right. What is the function of parietal cells? What do they do? Are they releasing an enzyme? Or are they doing something else? Yeah, so their job is to produce the HCl. So they release the hydrogen ions and the chloride, and that gives us the HCl. They also make intrinsic factor. Perfect. What does intrinsic factor do? Yeah, so intrinsic factor helps us with B12 absorption. I should put that on the slide. All right, what is bile and why do we need it? Is bile an enzyme? No, bile is not an enzyme, but bile is still important because what does it do? Yeah, it emulsifies fat. So it allows us to take these big fat particles, which we can see here, and then break them down into these smaller particles because these smaller particles are easy for the enzymes to work with because they have more surface area exposed. But bile itself is not an enzyme. Why is pancreatic juice alkaline? So why does pancreatic juice have a high pH? What's in it? Yeah, so we have bicarbonate in pancreatic juice. Why is that important? Why do we want to have bicarbonate in our pancreatic juice? Yeah, because what's going to happen is that we are going to release contents from the stomach. Those contents from the stomach are acidic, and we need some way to neutralize that acid. So the pancreas releasing the pancreatic juice with bicarbonate in it allows us to neutralize that acidity. That way we don't damage our intestines. There's other stuff in there, right? There's water, obviously, and then there's going to be a bunch of enzymes as well. But the bicarbonate specifically is to help us to neutralize the acid from the stomach. All right, what are circular folds and what is their purpose? So first of all, who has circular folds in our digestive system? Yeah, so the circular folds are going to be in the small intestines. Rugae are going to be in the stomach, the gallbladder, the urinary bladder, stuff like that. So they are going to be in the small intestines. What is the purpose of the circular folds? Yeah, so they slow down the movement of the food, which gives us more time for absorption. They also create some turbulence, so more stuff is bumping against the walls, which is perfect. And they also do the thing that the small intestines are great at, and that is increasing surface area, making it better for absorption. So all the stuff they do really enhances our ability to absorb stuff, and that's perfect because the small intestines are the rock stars of absorption. All right, urinary system. What are the four parts of the urinary system from superior to inferior? So, what is the highest up thing in the urinary system? Two of them. Kidneys, perfect. Then kidneys are going to produce urine and send it through the ureters. And then we're going to get down to the bladder where we store that urine. And then bladder leaves the urinary system through the urethra. So, kidneys are the most superior thing. They produce urine. Then the urine travels down a ureter to get to the bladder, which is for storage. And then the urine will leave the body via the urethra. Kind of cut off urethra there, but we can still see the organ. All right, describe the filtration membrane. There are three parts to it. If you were, I don't know, like sugar in the blood, and you were going to end up in the filtrate, what would be the three things you had to pass through? Yeah, so the three things we need to pass through. So the glomerulus is just a capillary, right? And so it's going to have the same kind of setup. So here is our capillary. So we're going to have to pass through the capillary endothelium with these fenestration pores. Then once we pass through that, we have to pass through the basement membrane, which would be the second thing. And then finally, we'd pass through the podocytes. And the podocytes have this slit diaphragm, which is made up of their foot processes. So the three things we'd pass through would be the fenestration pores of the capillary endothelium, the basement membrane, and then the slit diaphragm of the podocytes. So if you were a molecule of sugar and you were going to be passed into the filtrate to be tubular fluid, you would pass through a fenestration pore, basement membrane, and then the slit diaphragm. Oh, sorry. What cannot pass through? What would be something that could not be filtered by your kidneys? Yeah, big things. Red blood cells, white blood cells, big proteins, those kinds of things. So, what gets filtered are small things, because small things can pass through all of these three things. What can't be filtered are big things, blood cells, big proteins, that kind of stuff. All right, during glomerular filtration, what causes water and small molecules to move out of the blood and into the capsular space? What drives the function of your perfect kidneys, right? What drives the function of your kidneys is pressure. Pressure is what allows for stuff to go from the glomerulus to this capsular space. Blood pressure does this. Specifically, it's the pressure in the glomerulus, but we can just generally say blood pressure. All right, what are the three functions that we have in the nephron? So what are the three phases of urine production, or what are the three functions that we have in a nephron? We just saw one of them, right? We just saw filtration, which was going to happen in the glomerulus and allow for glomerular filtration. What are the other two things that we do? Yeah, we do reabsorption and secretion, right? If you are reabsorbing something, does that mean it will be in the urine or it will not appear in the urine? Yeah, so if you are reabsorbing it, you are keeping it, right? Reabsorption means you keep it. This is going to be like water, glucose, those kinds of things. What about secretion? If you secrete something, is it likely to appear in the urine or unlikely? Yeah, if you secrete something, it is likely to appear in the urine. You have taken it from the blood and put it into the tubular fluid. So reabsorption says, oh, I'd like to keep that, whereas secretion says, I'd like to get rid of this. Reabsorption, keep it. Secretion, get rid of it. And so, big picture, what this means is if you are filtered, if you are a molecule of glucose and you are filtered, you could appear in the urine unless you get reabsorbed. So, if you are filtered and reabsorbed, you get to stay in the blood. If you are filtered or secreted and do not get reabsorbed, you will be in the urine. You will get excreted. All right, so let's work our way through a nephron real quick and make sure we know all the processes. So we're first going to filter, like we just said, in the renal corpuscle with the glomerulus. Then after that, what is this first tubule that we're going to work through? After we get filtered, we are tubular fluid. What is the first tubule? Yep, the proximal convoluted tubule here. Then we're going to work through this thing. What is this thing? Yep, nephron loop. We're going to descend first, and then we're going to ascend, right? And then after that, we're going to be in this next thing right here. What is this thing? Yep, the distal convoluted tubule. And then we're going to pass through the collecting duct, but the collecting duct is not a part of the nephron. So filter at the renal corpuscle, work through the proximal convoluted tubule, the nephron loop, and then the distal convoluted tubule. Then after all that, we'll pass out of the renal papilla down here and into the minor calyx. All right, last thing is reproductive system. So what are the primary sex organs in male and females, and why are they considered primary? Or what are the gonads? Yep, testes and ovaries. And what do they do that nothing else in your body does? Gonads make what? Yep, they produce gametes. So their job is to produce gametes. They also do something that, other parts of your body do, make hormones, but they specifically make sex hormones. So the testes are going to be the male gonad, and the ovary is going to be the female gonad. So they produce gametes. Here's an egg. I can't show you a sperm in this picture, but use your imagination. And they also produce sex hormones. So there's the corpus luteum there. And then I don't have the testes picture is pretty vague. What is the structure where sperm mature so that they are fully functioning? Or you could think of this as like, where do sperm go to learn how to swim? Yep, the epididymis. So the epididymis has different sections to it, but this is the epididymis. This here is the vas deferens, and then here is the testis. What are the accessory glands of the male reproductive system, and what do they do? So there are three of them. Yep, the prostate is one of them. Perfect. Yep, we have the prostate, we have the bulbourethral gland, and then we have the seminal vesicle. Awesome. So the seminal vesicle, the prostate gland, and the bulbourethral gland, and all of them serve to do what? They basically all serve to keep sperm from dying, right? So sperm are kind of helpless, right? So sperm could die from a low pH. So they provide alkaline fluid to make sure low pH doesn't kill sperm. Sperm have to have their energy provided to them in a simple form because they can't really break stuff down very well. So we supply them with fructose, and they also provide some mucus to make the semen mixture and to make sure it's the right viscosity. So all of these things are just helping sperm to not die before they even get out of the male reproductive system. How does a newly released ovum get to the uterus? So during ovulation, an egg gets released. How does that egg go from the ovary to the uterus? Yeah, through the fallopian tubes. How does the egg get into the fallopian tube? Yeah, perfect. So the fimbriae, right? Those fimbriae are going to create a current. So here we can see the fimbriae here. They are going to create a current that draws the egg into the fallopian tube. Then the fallopian tube will be the pathway that the egg travels along to get into the uterus. If the egg gets fertilized in the fallopian tube, then it could implant somewhere in the uterus. And then if not, then it'll just pass on. So in the fallopian tube, there'll be other things to help move the egg along. This would be things like cilia beating in a rhythmic fashion, right? There'll also be some peristaltic contractions, not like aggressive peristaltic to like crush the egg, but enough to keep it moving. All right, where does gonadotropin releasing hormone come from? And what does it tell us to do? So if we think about the endocrine system that we talked about before, gonadotropin, sorry, that's wrong text. Ignore that. So gonadotropin releasing hormone is going to be coming from the hypothalamus because the hypothalamus tells the pituitary what to do. So GnRH is going to then go to the anterior pituitary and tell us to release LH and FSH, hormones from the anterior pituitary. And these are going to tell us to do a couple of different things. LH is going to tell us to release hormones. In the case of the female reproductive system, it's going to say produce things like estrogen. In the male reproductive system, it'll say things like produce testosterone and release it. FSH is follicle stimulating hormone, so it's going to say produce or mature gametes. So in the female reproductive system, it's going to say to produce and release, not produce, sorry. It's going to say to mature and release the whatever is the most mature oocyte. And then that's going to lead to ovulation. So LH is for sex hormones, FSH is for gamete maturation and release, or in the case of the male reproductive system, gamete production. So GnRH comes from the hypothalamus, and that's because the hypothalamus tells what the pituitary what to do. Then the pituitary produces LH and FSH. LH is for hormones, FSH is for gametes. All right, when would an oocyte complete meiosis II? Last question. Yeah, so when we think about meiosis, the male reproductive system just goes through meiosis completely to produce sperm, but the oocytes only complete meiosis II after fertilization. So way down here, the only oocyte that's going to complete meiosis II will be the one that gets fertilized. So that's going to be a stark difference between how the male and female reproductive system produces its gametes. The male reproductive system just goes through meiosis I, meiosis II over and over again. Female reproductive system has stopping points, and the big one for completing meiosis II is fertilization. All right, so that is all the prepared questions I have for you. You have just a couple of days left until you're done with the semester, so I'm not going to keep anyone extra long. I know I went over for a few minutes. If you have more questions, I'm happy to go through anything that you want. And if you don't have any more questions, then go and good luck on your final exams. Let me know if there's anything else I can do. If you come up with questions, email me, stop by office hours, let me know. Otherwise, go enjoy the rest of your week, get through finals, and have a spectacular summer.
I have a question
Yeah, absolutely.
on it was one of the slides back when it said releases oxytocin and something else.
Vasopressin. So the posterior pituitary is going to be responsible for oxytocin and vasopressin.
You said that's the anterior pituitary gland.
Posterior pituitary.
Oh, posterior.
Yeah, so the anterior pituitary is going to be The mnemonic I use for that is flag top. So the posterior pituitary is going to be vasopressin, antidiuretic hormone, which is the same thing, and oxytocin. For the anterior pituitary, the way I remember it is to remember flag top, and the F
Yeah.
is for FSH, the L is for LH, the A is for ACTH, the G is GH, the T is for TSH, the O is omit, it's not a hormone. There's just not enough vowels in this for it to be a good mnemonic. And the P is for prolactin. So FSH is follicle stimulating hormone, LH is luteinizing hormone, ACTH is adrenocorticotropic hormone, GH is growth hormone, TSH is thyroid stimulating hormone. So anterior, flag top, posterior, oxytocin and vasopressin.
Okay.
That makes sense now?
Yes, thank you.
Yeah, of course. Yeah, so Sertoli cells, also called sustentacular cells sometimes, they're just babysitters for sperm. So the Leydig or Leydig cells, I'm not 100% on the pronunciation, they are responsible for saying, or for producing testosterone, whereas the Sertoli cells are just responsible for making sure that the sperm are progressing through development at the correct correct rate and pace. They also just kind of do like sperm babysitting, making sure they have nutrients, making sure they're taken care of. So Sertoli cells are basically just sperm babysitters, because otherwise you just have a bunch of cells that don't have a full set of genetic information. So someone's got to watch over them, and that is the Sertoli cell's job. So Sertoli, sperm babysitters, Leydig cells are producing testosterone. Yeah, have a good one. Let me know if you have more questions.